Okay, hi everyone. Thanks for joining yet another RISE session. So today we have a very special guest, Dr. Lasya Sreepada, uh, and she'll be talking about her thesis work on epigenetic signatures of aging and neurodegeneration in Alzheimer's and related dementias using machine learning. This will be a bit different from my usual talks. It will be more clinically focused, so it will be nice to uh, get a get away with get away from methods related papers and try to see something more biological and clinical. So Dr. Lasya Sreepada is a data scientist at BrainSpec in Boston. She earned her PhD degree in bioengineering and a master's in data science from the University of Pennsylvania. And she holds a bachelor's degree in neuroscience from Yale. Her doctoral research centered on integrating neuroimaging, multiomics, and AI and machine learning models to uncover biological mechanisms of aging and neurodegeneration in Alzheimer's disease. Uh, yeah, Lasya, take it away.
Yeah, thank you so much, Pulkit, for that very kind introduction. And it's great to see you again after a little while. And it's great to be here with everybody to share a little bit more about my thesis work that I just recently completed in late July. So again, I'm Lasya. I welcome any interruptions throughout my talk. I recognize that this is a little more applied and application oriented, especially within the clinical and biological domain. So I'm happy to have any questions from you throughout the talk as well as at the end during the discussion. So with that, I'll just get into it. So I wanted to give a little bit of background about kind of really take a big step back and talk about what is the realm that we're focusing on here, and that's aging, which is underlying many changes that occur physiologically throughout the body with time, right? And we see a lot of these changes occur in the body at the cellular, molecular, and physiological level. Of course, I'm interested in epigenetic alterations, but there's so many factors that we can consider when studying aging. And of course, these processes are part of normal or healthy aging, but they can also be pathological when they go awry. And these pathological processes can contribute to a wide variety of disorders, including autoimmune disorders, cerebrovascular disease, motor neuron disorders, and neurodegenerative diseases, which is my area of interest. So we know that aging and disease actually affect individuals very differently. And I have this illustration here on the left-hand side, which shows you a picture of two identical twins. And, naturally what that means is that they're the same age, but someone might erroneously predict that or guess that the individual on the right-hand side of this image is older than the individual on the left-hand side. And we know that this isn't the case because they're identical twins. But what this illustrates to you is that various factors, including life exposures such as sun exposure, smoking, drinking, or nutrition, exercise, all of these are lifestyle factors that could affect the biological age processes in an individual that may make them look or feel or behave as if they are older or younger than their given chronological age. So this is also the case with many diseases, and in particular Alzheimer's disease, where we have a wide range of disease onsets. You see these two peaks here. Most often Alzheimer's onsets around the age of 75, and this is what's considered late onset Alzheimer's disease, and it's the most common form. But there's also early onset Alzheimer's disease, which has a different distribution and often onsets around the age of 65 or even earlier. So one major aspect of my work is to tease apart, besides age, what are some of the factors that are differentiating the disease types in early and late onset, because we actually do know that they have very different presentations in individuals. So the concept that aging and disease affect individuals differently is known as heterogeneity in our field. And this naturally leads to the question, how can we explain heterogeneity? And one framework is, of course, genetics and genomics. The DNA that makes up every single one of our cells is providing kind of a blueprint for how our bodies work and how they develop and age. But this is only part of the picture. Like I mentioned on the previous slide, we have various exposures and lifestyle factors that contribute to how quickly or how slowly somebody is aging relative to their to the time elapsed since birth. And a lot of these are socioeconomic or systemic factors, including education access, economic stability, healthcare access. And these factors can also influence, like I mentioned before, lifestyle factors such as nutrition and exercise that are somewhat in your control, but are being influenced by outside factors as well. And finally, this leads to the field of epigenetics and epigenomics, which is again the focus of my talk here. And I find epigenetics a very interesting data modality to work with because it accounts for both heritability, which we see in genomics and in genetics, but it also accounts for exposure and lifestyle. We know that epigenetics, which is the study of variations in gene expression that don't involve changes to the DNA sequence, it's affected by lifestyle factors. And so, for example, DNA methylation is one type of epigenetic marker or modifier that controls or regulates the expression of genes that are either upstream or downstream of this marker. And the level of methylation at each of these, what are known as CpG dinucleotide sites, changes based on our environmental exposures and of course other factors, but I'm just harping on the environmental exposures part to really illustrate that this is a dynamic measurement that we can assess throughout the lifespan, especially when we're acquiring this data in blood or other peripheral tissues that can be readily accessible throughout life. And so you see if you remember back in organic chemistry, if you ever took that class, what's happening is basically there's a methyl group that's attached to the cytosine base pair at these cytosine guanine dinucleotide pairs throughout the genome. And recently, there have been many statistical and machine learning approaches that use epigenetic profiles to estimate robust measures of biological aging. Now that we've sort of covered what are some of the frameworks with which we can explain heterogeneity in individuals, we also want to know what's happening in the organ of interest. And for me, I mentioned that I'm interested in neurodegenerative diseases and the brain. So we want to be able to relate these explanations, such as epigenetic factors or genetic factors, to the actual phenotypes, or what is actually, how can we measure the effects of these changes throughout the body. So neuroimaging provides us a window into the aging brain. And we see here that, in healthy aging, this is perhaps what a structural MRI scan would look like. And I also have 3 other examples of how you can visualize the effects of Alzheimer's disease on the brain in varying forms. So I mentioned typical Alzheimer's disease, which is often late onset and occurs at 75 years of age or older. You see a lot of atrophy or degeneration in the brain, in the frontal lobes, in the parietal lobes, and the temporal lobes as well. But there are also other presentations of Alzheimer's disease in which you have more predominant atrophy in either the temporal lobe, the medial temporal lobe, or in cortex or the frontal regions of the brain. And so these are very heterogeneous presentations of Alzheimer's disease. And as many of you are familiar with, and maybe you've designed tools to do this, we're able to parcellate the brain into anatomically defined regions, or even functionally defined regions as well. In this case, I'm focusing on structure. We can create machine learning methods or deep learning methods that are able to parcellate the brain into regions of interest. And we can quantify different aspects of these parcellated regions. So it could be brain volume, it could be cortical thickness, any sort of quantitative measure that is providing us with more information about what is actually going on in this region of interest. And in particular, in my work, I'll be focusing on, excuse me, the hippocampus, which is part of the medial temporal lobe. You can see it here in the structural images. And the hippocampus is a structure in the brain that is noted to be related with memory and is most often implicated in aging and neurodegenerative diseases such as Alzheimer's. So I mentioned various data modalities that we can use to explore aging and neurodegeneration, especially as it relates to Alzheimer's disease. And what I want to highlight about these data types is that they're both very high dimensional, both epigenomics and neuroimaging. So we need appropriate tools to be able to glean patterns from these data sets that are very high dimensional, and perhaps we have greater number of features that we can extract from these data sets than we have samples, right? So machine learning, as you are all familiar with, is a framework with which we can design or write programs for a computer and enable this computer system to learn from data sets and improve their performance on specific tasks without being explicitly programmed for each new data instance that we're providing the system. And how I view machine learning is as two fundamental considerations. One is the data wrangling or the inputs that you're providing to your machine learning model, as well as the model design, right? So they're both, in my view, equally important in designing a very accurate and meaningful or interpretable model. So of course, a big part of my work and my talk will be focused on how we performed biologically meaningful feature selection, or part of the data wrangling process, and also the type of model that we chose in terms of regularization or interpretability, but also focusing on how optimized the fit of this model is to our data set and whether we are underfitting or overfitting, and really trying to get closer to this middle part where we're really capturing the relationships between the variables that we're interested in without fitting too much noise. So this leads to the central question and the hypotheses that I'll be testing and answering during my talk. So the central question is, how does blood-based epigenetics associate with neurodegeneration and modulate heterogeneity? And this central question was broken down into two aims. The first being understanding the role of biological age acceleration, or that discrepancy that I was mentioning between an individual's chronological age and their actual biological rate of aging. So how does biological age acceleration, defined using blood-based epigenetic clocks, which are machine learning methods that can predict biological age, how does this influence heterogeneous neurodegenerative patterns in Alzheimer's disease? And the second question is a more data-driven approach, looking at the entirety of the epigenome and not just a selection that are used in these clocks. How do blood-based epigenome-wide markers modulate aging and region-specific neurodegeneration in the hippocampus? So with that, I will get to the first aim of my talk. So here I want to go back to the concept of heterogeneity and atypicality in Alzheimer's disease, and actually really explain a little bit more about what atypicality is. So I mentioned previously in heterogeneity, aging and age-related processes are affecting individuals differently. And what we see in Alzheimer's disease is that in some individuals, we have greater involvement or atrophy in cortical structures, which are denoted here with these colored dots, and labeled with the downstream cognitive functions that are affected as a result of atrophy in these areas. And then, of course, there's also, again, the medial temporal lobe, of which the hippocampus is a part, which is typically associated with changes in memory. So we have individuals that have greater or lesser involvement of either cortical or medial temporal structures. And that's an aspect of heterogeneity that we're trying to understand. And I'm bringing back this image from the introduction to the talk, where I pointed out that there's a typical Alzheimer's disease, but there's also MTL predominant, medial temporal predominant, or cortical predominant versions of Alzheimer's disease. So what I've done in my work is I've kind of characterized these ends of the spectrum as, greatest atrophy in the medial temporal, or greatest atrophy in cortical structures as being two ends of a spectrum of atypicality in Alzheimer's disease. And what we're trying to do is understand how can we explain where along that spectrum an individual might lie. Another thing that we know is that age is imperfectly associated with disease presentation. So, I mentioned we have this late onset form of Alzheimer's disease that often onsets around 75 years of age. But we also have various other, ages of onset. And these ages of onset are actually known to be associated with the typicality of Alzheimer's disease, or where we see the most atrophy. So actually, the late onset typical Alzheimer's disease is most often associated with a medial temporal predominant atrophy pattern. But we also have early onset Alzheimer's disease, which, you saw the distribution on an earlier slide, that individuals contract this disease perhaps 65 years or under. And while early onset Alzheimer's disease is often associated with atypical presentations or more cortical presentation, this isn't always the case. And here's an image that can help illustrate that. So on the top here, in this top row, we have some structural MRI images acquired from an 81-year-old who has an amnestic presentation of Alzheimer's disease, meaning that they have a memory predominant, so amnestic presentation. And we know that that's associated with greater atrophy in the medial temporal regions. Below, we also have a 57-year-old, so definitely comes under the early onset spectrum, who also has an amnestic presentation. So this is a simple example that indicates to you that simply age of onset, or greater than 75 or younger than 65, isn't really sufficient to capture where along that atypicality spectrum, both from a structural perspective as well as a downstream cognitive perspective, where an individual might lie. And this is something that we're trying to probe. So again, while older individuals often have typical amnestic presentations and younger individuals tend to present atypically, this isn't always the case. So we have to go beyond just chronological age to further answer this question. And this leads to the concept of biological aging, right? And we hypothesize that biological age acceleration, or again, the discrepancy between an individual's biological age and their chronological age, might modulate that typicality axis beyond just chronological age. And so this kind of gets into a little bit of the machine learning aspect of this work. So there is a method, and these are increasingly popular these days, called an epigenetic clock, which is able to predict biological estimates of age using the epigenetic profiles of an individual. So they're looking at the DNA methylation percentage at many of those sites across the genome. And what this helps us do is, once you predict a measure of age using the DNA methylation or other epigenetic modifier, you can then look at, again, the discrepancy between the predicted age and the input or the chronological age. And an individual might be accelerated or in red, or decelerated and in blue. And this gap here, or the discrepancy, is what we call biological age acceleration. And that measure is, or residualized measure basically, is of interest to us in understanding an individual's rate of aging and also their disease presentations. So this on the right-hand side is actually my data. And so, you see here again that in individuals for whom we had DNA methylation data, we estimated their biological age using a variety of these epigenetic clock machine learning models. And this is the data distribution that we had got, right? So on the y-axis, I'm plotting the DNA methylation age prediction, and on the x-axis, I'm plotting the actual chronological ages of these individuals. And what you see is that both estimates are correlated. So of course, you would expect that somebody's biological age is related to their chronological age, but that there is this variance, right? And you can see that these individuals in red here are considered to be biologically accelerated or older than their given chronological age, and similarly, those in blue are younger. So while you see a correlation between biological age and chronological age, we see this variance and discrepancy, which allows us to measure this age acceleration. So I'm going to then talk about the actual study design. So how did we begin to address this question? Many of you might be familiar with the Alzheimer's Disease Neuroimaging Initiative dataset, which is what we used here. And this dataset is pretty useful in this context because it has both structural neuroimaging and DNA methylation data from which we can estimate biological age. So a lot of, I'm going to point out throughout my talk some of the challenges that we encountered, and a lot of it has to do with sample size, which I could trace back to availability of data sets that have both data modalities. So just something to keep in mind. But we used the ADNI dataset, and we separated our cohort into cognitively unimpaired individuals or symptomatic individuals with either mild cognitive impairment or Alzheimer's disease as defined by clinical consensus. And for each of these cohorts, we were able to compute or estimate biological age and neurodegenerative pattern as follows. So for the biological age aspect, we have our DNA methylation profiles in individuals. We applied these epigenetic clock models to estimate biological age, and then we computed the biological age gap or age acceleration measure that I just explained on the previous slide. On the neurodegenerative pattern side, we have structural T1 MRI for all of these individuals. We standardized these measures using essentially z-scores that are accounting for age and sex. And then we computed an index, a novel index that I'm introducing in this work here and that you can read about in this paper, Neurobiology of Aging. And this index is able to quantify that axis of atypicality that I was mentioning. And we're ultimately performing statistical analysis to relate these measures of biological age acceleration with the CoMeT index that's quantifying atypicality in Alzheimer's disease. And I'm going to break down CoMeT for you. So CoMeT stands for the Cortico-Medial Temporal Index, and you can understand why on the first slide where I was of this aim when I was explaining the two ends of the spectrum, right, where we see the most atrophy. So in blue here, we have the cortical structures that are most frequently impacted in Alzheimer's disease, or also known as the cortical signature of Alzheimer's disease. And in red, we have regions that are considered part of the medial temporal lobe, plus the hippocampus and amygdala, which I wasn't able to include here on this figure, but they're also included in that signature. And what we did is we computed basically, again, a z-score or a w-score, if you will, depending on your choice of terminology. We were able to compute these standardized measures of structure. So in most cases, we used cortical thickness. For the hippocampus and the amygdala, we looked at their volumes. But ultimately, because we were standardizing these scores, we were able to develop a composite score for each like primary region that we're interested in, which was, again, the medial temporal lobe and the cortical structures. And then we just took the simple difference of these composites, right? So we did cortex minus medial temporal lobe is equal to CoMeT. And this obviously resulted in values that are similarly on the same scale as a z-score, so that they're interpretable in that way. And the way to interpret CoMeT is that a more positive value indicates more MTL predominance, and a more negative value indicates cortical predominance. And then this is just, the sign is just because of the way that we constructed CoMeT, which is cortical minus medial temporal. And the reason we did a difference as opposed to a ratio of these two composites is to preserve that sign, which would help us interpret, which set of regions is actually contributing to the value of CoMeT, and that would help us understand where individuals lie along that cortical medial temporal spectrum. So in this first slide, I'm basically just confirming that our computation of CoMeT is in alignment with what we see in the literature. So as I previously mentioned, early onset Alzheimer's disease tends to associate with greater cortical involvement or greater atypical presentation of Alzheimer's disease. And this has been established in the literature. And in this figure here, I'm showing you that in the symptomatic individuals, we see a similar effect where younger individuals who are below 65 years of age have a lower or more negative CoMeT value than individuals who are older than 80. And this is a significantly different relationship between the means of these two different distributions. Whereas in the cognitively unimpaired individuals who are used as a reference, the mean of CoMeT is around 0, and this is what you would expect. So this basically aligns with our understanding that younger individuals tend to have more cortical predominant presentations, while older individuals have more MTL predominant presentations. But so this is an excellent validation of CoMeT, but of course we want to still address that question that we had of, well, we know that chronological age can only capture so much of the variance that's explaining this heterogeneity in individuals. What else, right? And this is where biological age comes in. And so just as a reminder, we computed the biological age acceleration measure using the biological age estimates that we acquired from the epigenetic clocks or machine learning models. And then we basically grouped individuals into accelerated or decelerated groups, using a threshold of 0.5 standard deviation above and below the mean of this distribution to designate these two groups. Now, you might ask me about my choice of threshold here. We actually did vary this threshold up to about 1.5 standard deviation above and below, and even smaller, 0.25 above and below. This threshold was chosen to optimize for the sample sizes across these 3 different groups. So we tried to ensure that we had approximately similar numbers of individuals that fell in each of these categories. So we're optimizing for sample size, but also for effect size between the accelerated and decelerated individuals to really look at the extremes of the spectrum. But there's so many, acknowledge that there's so many different thresholds that you can use, and this is just our reasoning for why we did it this way. So indeed, we noticed that biologically decelerated individuals, or folks who are younger than expected than their chronological age, also show greater cortical involvement, a greater cortical structural involvement relative to accelerated individuals who are older than expected for their chronological age. And this is, again, a significant difference between the means of these distributions, and also replicated in a continuous analysis where we plotted CoMeT against biological age acceleration. So in conclusion, in this work, we demonstrated that CoMeT, or the cortico-medial temporal index that we defined, is able to quantify the relative involvement of cortical structures to medial temporal structures involved in Alzheimer's disease. We validated the use of CoMeT by showing that younger chronological age is linked to more cortical presentations of Alzheimer's disease. And then we expanded this validation with a novel finding that decelerated biological age, or being biologically younger than your chronological age might indicate, is also linked to more cortical presentations of Alzheimer's disease. And in conclusion, overall, we can derive from these findings that biological age acceleration modulates the typicality of Alzheimer's disease beyond the known effects of chronological age. And some of the work outputs for this project are a paper that was just published in Neurobiology of Aging in June, and as well as various conference posters, platform presentations, a chapter in my dissertation, and code to compute CoMeT is available on my GitHub. So coming back to the central question and hypotheses, we asked the question, how does biological age acceleration defined using blood-based epigenetic clocks influence heterogeneous neurodegenerative patterns in Alzheimer's disease? And this is what we found. Biologically decelerated individuals tend toward cortical predominant neurodegenerative patterns, while biologically accelerated individuals demonstrate more typical medial temporal predominant patterns. And now we can move on to looking at epigenome-wide markers. So not just, one thing that I perhaps didn't mention is that in epigenetic clocks, typically you look at maybe 300 to 1,000 sites or features across the epigenome and look at the methylation patterns at each of those sites and use those as predictors of your biological age estimate. And that's great, but sometimes we're interested in looking at more data if we have it. And so in this second aim, we'll look at epigenome-wide markers and see how they might modulate aging and region-specific neurodegeneration in the hippocampus. So just again, to recapitulate, we can acquire DNA methylation profiles in a population from various tissues, including brain tissue, liver tissue, but here we're focused on blood tissue because it is readily accessible, not invasive, relatively, and is also, we're able to acquire it longitudinally throughout the lifespan and observe these dynamic changes in methylation of these different markers throughout the genome and throughout the lifespan. And from these DNA methylation profiles, we can estimate or relate these measures to things like chronological age, cellular age, biomarkers that are associated with clinical aging, as well as other exposures throughout the lifespan that I had previously mentioned. And what I want to note is that there are two different ways that we can analyze these epigenome-wide profiles, or so the full set of data that we have here. And one is a more traditional statistical approach known as an epigenome-wide association. It's basically like an F-test conducted at so many different sites or features across the genome. So this is basically multivariable testing. And you may have actually heard of a genome-wide association, which is very similar, except instead of using epigenetic markers, you're using single nucleotide polymorphisms or SNPs across the genome. And in this case, it's in fact the epigenome and methylation markers throughout the epigenome. So this is a very traditional statistical approach that you can use to analyze the entire epigenome. Or you can also use data-driven machine learning methods, which are known to be able to handle large multidimensional data sets. So you can see in this figure here, you have your DNA methylation profiles, which are in cream as an input layer. And there's kind of two ways to do this. So the first way, in the bold arrow, is really what I focused on in my thesis. And I'm interested in pursuing the deep learning aspect further now that I'm sort of continuing to work on the paper after my defense. But basically, your inputs are these DNA methylation markers, and your outputs are, any measure of biological age, which in this case is DNA methylation derived age. You can use any kind of penalized regression to get from this input to the output, or you could have multiple hidden layers in any kind of deep learning based approach to really learn associations sort of among these features as well, and develop perhaps latent space that captures a lot of the information that's shared between chronological age and biological age, for example, to ultimately derive this final output. So these outputs could be any of the following. You could, in traditional epigenetic age models, we're predicting chronological age, and that's the Y variable that we're training on, that we're trying to predict. You can kind of appreciate, though, that if you are training a machine learning model to predict chronological age, and your methods of estimating the value or the performance of that model are accuracy driven, the more you estimate chronological age, or the better you estimate chronological age, you might actually be suppressing signal that's associated with biological changes, right? So there is that sort of push and pull between how accurate do you want a model to be when predicting chronological age versus how much of the biology do you want to be capturing, right? So that's definitely a tug of war right now in the field. So traditional models have chosen to predict chronological age. You could also predict disease risk and mortality. So these are age-related phenotypes that we can predict using this data. And finally, you can actually predict those phenotypes themselves. And, to that extent, something like hippocampal volume or various brain volumes are kind of proxies, very noisy proxies for age itself, right? Because we know that in even healthy aging, we see atrophy in these brain regions. So kind of these brain volumes are a proxy for age itself. So in this case, I'm actually focusing on predicting hippocampal volume from DNA methylation profiles, and trying to see what we can understand from an epigenetic perspective as to what factors are driving changes in hippocampal volume, especially from the epigenetic perspective. So just again, motivating the use of blood-based epigenetics as a surrogate for brain changes. You might ask me, well, if you can acquire DNA methylation from a variety of tissues, why didn't you acquire it in the brain? Why are you looking at blood? So first of all, we know that there are associations between blood DNA methylation and Alzheimer's disease. So this provides basically a basis for us to even consider this data modality. But also, we know that blood, as I mentioned, is easily accessible and you can acquire it throughout the lifespan. If we wanted DNA methylation data from the brain, this would necessitate a post-mortem analysis, and we wouldn't really be able to see how these dynamic changes in DNA methylation are influencing neurodegeneration in the disease process, right? There's some sort of bias that we would encounter when we're looking at individuals who unfortunately passed away from the disease as a result. And so in this way, using blood-based epigenetics allows us to study the disease in vivo. And I previously mentioned that, the traditional statistical approach to study DNA methylation or the epigenome is to use a, epigenome-wide association study in which you test the association of DNA methylation at many, many sites across the genome or epigenome with the phenotype of interest, and in this case, hippocampal volume. This study has actually been done. This is real data from this paper, Jia et al., 2021, Molecular Psychiatry. And this study shows that indeed there were actually 340 significant sites across the epigenome. Again, those are known as CPGs, that are significantly associated with hippocampal volume. And this pie chart is actually showing that not only are 71% of those significant CPGs associated with the hippocampus, but we also have among these CPGs, they were also associated with other regions, other subcortical regions, including the thalamus and the nucleus accumbens, and some that were significantly associated with brain volumes of all three. So what this shows us perhaps is that maybe there is some sort of significance between DNA methylation changes and structural change in the brain. So this motivates us to then use machine learning methods that might help us capture more variants or discover new patterns that we're unaware of using a more data-driven approach. So I previously mentioned that epigenomics and also neuroimaging suffer from this curse of dimensionality. And just to really highlight how bad it is, in DNA methylation assays, this is the chip that we use to perform the methylation assay. It's often from a company called Illumina, and this is the chip. It's able to measure DNA methylation at almost 1 million sites across the epigenome, right? So that's a lot. And in contrast, we have around 225 individuals in ADNI that have both epigenetic data and neuroimaging data, right? So I mentioned that the multimodal data requirement is a challenge that we needed to overcome in terms of accruing a good sample size. So naturally, this, the feature space is orders of magnitude larger than the number of samples that we're able to use to train any model. So what we realized is that this necessitates the use of some kind of feature selection method prior to training any kind of model to predict epigenetic age or biological age. And interestingly, this is actually not very common in the field so far of epigenetic clock development. It has only been introduced into the pipeline more recently. Traditionally, people had just used some kind of linear model and used data that actually, in older versions of the chip was maybe only acquiring 30,000 or 40,000 features. So as the technology is improving and we're able to more accurately quantify epigenetic data at much larger number of sites, we also need to advance the field in terms of how we perform feature selection to pick the epigenetic features that might be most related to predicting our phenotype of interest. And in this case, that's hippocampal volume. So here I have a graph that shows some of the supervised feature selection methods that I had tried to optimize in my work. So of course, we can use a filter-based feature selection method or embedded using some kind of regularization penalty on the model. So in the filter-based method, we computed these values for each of these CPG sites across the epigenome in association with hippocampal volume. And then we ranked all of these features based on these metrics and selected the top N features, say the top 100 that are strongly correlated with hippocampal volume. So this was a filtration-based method. And of course, the embedded method actually ended up being our baseline. And we chose an ElasticNet model to combine both L1 and L2 regularization in our model. So traditionally, actually, ElasticNet itself has been used as the base model for any epigenetic clock. And so this was kind of our baseline. And then we also added on this feature selection module that helped us cull our feature set into something a little bit more manageable and smaller, especially given our sample size, which I'll highlight here. For our training set, we had ADNI, and for our testing set, we actually had the aging brain cohort, which was acquired at the University of Pennsylvania. So it was a local cohort, and I believe has not been published widely yet, which makes it exciting. So we have what I want to note here is that between these two data sets, we actually did have some protocol differences in terms of the MRI acquisition, as well as the DNA acquisition. So some of these protocol differences proved to be somewhat detrimental to combining these data sets into a single data set that's harmonized that can be used for training. And so that was another challenge when working with multimodal data is, how compatible are the data sets that we're working with. But, just to show you the data that we're working with and the size that we have. So in terms of the study design, again, we started with machine learning, in which we trained and tested machine learning models using these two data sets and identified converging epigenetic signatures. Next, we employed a variety of bioinformatics techniques to perform various analyses such as gene set enrichment, gene ontology, and pathway enrichment that helps us make sense of the features that were selected using our machine learning models or weighted with non-zero weights by the model. And finally, we have our biological interpretation component of the work where we took a look at, from the bioinformatics analyses, what can we understand about the genes, the CPGs, and the biological pathways that are emerging as significant, and how can we use that for perhaps drug target discovery or understanding disease mechanism, which will help us hopefully in the future better treat and better manage diseases such as Alzheimer's disease. So this is adapted from Doherty et al. From BMC Bioinformatics in 2023. We used a two-stage machine learning pipeline to identify epigenetic signatures, basically sets of features from the epigenome that are associated with hippocampal volume, which is the testing or the variable that we're trying to predict the output. So in this internal validation stage, we used a 5x3 nested cross-validation approach where feature selection using those ranking method was performed within the training set. And within that, we also performed the inner cross-validation loop for hyperparameter tuning of the ElasticNet model. And once we optimized this model at this stage, we basically had to select not only which feature selection method yielded the best final performance, but also the number of features chosen based on the ranked lists. So for example, if we had 900,000 ranked features based on a Pearson correlation, was it optimal to select the top 20,000 features, the top 1,000, top 100, et cetera, et cetera. And so we did a very systematic estimation of what is not only the best feature selection method, but also the best subset size. And then once we acquired, or once we did that experimentation and decided what the best protocol would be, essentially, we applied that optimal ranking and subset, excuse me, to the full training data, which was ADNI. And then here, instead of partitioning the data set again, we did a tenfold cross-validation to perform hyperparameter tuning for that elastic net model. And finally, we applied this fully trained model to the independent test set, or ABC. Now, kind of getting into the results here, as expected, we see that feature selection improves the prediction of biological age over the baseline methods. So you can see that in a baseline method where we used only ElasticNet without a prior feature selection model, we see that the test correlation here is 0.5, whereas one of the best performing methods with these ranking-based feature selection methods was 0.7, the test correlation. So what this shows us is that implementing this feature selection module prior to training a model to predict biological age is indeed effective at improving our accuracy. So what we then see, this was kind of a, yes.
Right. There's a question for you in the Q&A if you want to read along.
Okay, great, thanks. Let me take a look. So it says, thanks for the wonderful talk. It seems that there's a need to curate sufficiently large data sets to validate your methodology. Is it difficult to curate such data sets, and are people curating such data sets? Yeah, thank you so much, Yashar, for this question. Absolutely, it is difficult to curate really large data sets that integrate multimodal data. And indeed, there are people that are curating such data sets. I know of a group actually at Penn that's doing this. And even the Enigma consortium, for example, there are groups that are integrating imaging data with multi-omic data, whether it's genomics, epigenomics, spatial transcriptomics, proteomics, whatever omics you can think of. The challenge is, again, as I mentioned, standardizing the values that we are obtaining, whether it's from an omics perspective. There are batch effects with acquiring data based on the chip. So there could be changes in the protocol that each site has. There could be differences just in human error in terms of how somebody's performing the assay. And of course, for imaging, as many of you may be familiar, there are changes in scanners and protocols, and even field strengths and capabilities of these technologies as time progresses. And so all of these factors make it difficult to pool together very large data sets from across the world. However, there are some methods that have been used to mitigate this. And in fact, I have as well. I just didn't mention it here, but methods that such as CoMeT or empirical based methods that are used for statistical harmonization are common. But in general, I think folks like the Enigma consortium and other groups as well that may be doing this, I think a lot of them are coming together to develop standardized protocols for data acquisition so that this kind of pooling is facilitated and makes it easier to acquire really large Ns that are needed for more heavy deep learning work like this. Thank you.
I have a question too.
Yeah.
So I was just wondering if my understanding is correct. Much of the work that you presented is cross-sectional, based on cross-sectional data. I was wondering, have you thought about using any kind of longitudinal analysis, or how could it be integrated or be helpful?
Yeah, that's a great question. And you're absolutely right. A lot of the data here that I'm using is cross-sectional. I think longitudinal data would be incredibly useful for basically looking at, I think longitudinal neuroimaging might actually be more useful than the longitudinal epigenetic data. I actually looked at longitudinal changes in methylation measures. In ADNI, they have multiple acquisition points. And we see that particular data modality is very noisy, and measuring methylation levels, it kind of averages out to the same thing over 3, 4, 5 years. And so it's unlikely that changes in the methylation can be reliably detected to assume or infer effects on something like imaging. But it would be very interesting to see how far out from an image acquisition, if we acquire DNA methylation, how far out can we predict atrophy or decline in a certain brain volume or regional brain volume. And I think this would provide a very useful translational biomarker for screening or for staging studies. And I think in the future, integrating this kind of temporal data would be really exciting for the field, especially as we move towards precision medicine.
Thank you.
Yeah, of course. So thanks for the questions. So continuing back where I paused briefly, in this slide, what I'm showing you is that our framework, our machine learning framework using the nested cross-validation is effective to predict chronological age, which has been done reliably using traditional versions of epigenetic clocks. And so we see that when we use ADNI to train our model and test in ABC, we see a correlation of 0.74. We also tried reciprocal training just because we had small data sets. And so we wanted to see, if we would see the same result if we trained in the opposite data set and tested in the original one. And we see something very similar. So what this indicates to us, again, is that this validates our machine learning pipeline functionality. And we know that age is associated with epigenetic or DNA methylation-based age. So this makes sense. So moving on to the prediction of neuroimaging measures as opposed to age, we see a little bit of difference between the performance in the internal validation stage and the testing stage. And this is probably expected, again, because of that sample size. So I'll point out here that there is some overfitting and really great performance in the the training set, where, using the Pearson R-based feature ranking method, we achieved, about 0.8 test correlation in our internal validation data set. But this didn't replicate when we tested in the independent test set. So you can see here, it's pretty poor test correlation, where, in the ABC data set, we see a 0.06. And when we test in ADNI, having trained in ABC, very similar, we see a test correlation of 0.05. So by doing this reciprocal training, what we show is that this isn't an artifact of any specific data set. And perhaps what's happening is that, we don't have sufficient data to reliably predict imaging in a different data set. One question that might arise is, does this have anything to do with the imaging? Is there a difference in the hippocampal volumes of ADNI relative to ABC? And that is, in fact, no. We looked at the distributions of hippocampal volume in ADNI and ABC, and we found no significant difference in the means of those two distributions. So there is something else going on that is limiting our ability to generalize the model trained in one data set to another. One thing that we did to try and probe at that question is see, well, maybe the composition of the data sets, including race or other factors that make the cohorts different from each other, is contributing to this difference. And that definitely might be, because when we tested the ADNI trained model in just ABC individuals who identified as white, we noticed that the test correlation basically doubled, right? So it's a 0.14. It's still not a fantastic correlation, but this shows that definitely race or race-associated biological changes might be having a role here, and we definitely think that that's going to be on the methylation side. There have been studies that show that, for example, Black or African American individuals have consistently more accelerated ages than other individuals of other races. And this is not, we need to be clear here that it's not a function of race. Race is basically a proxy for other biological processes that are a result of, poor nutrition, socioeconomic status, and access to education and things like that. So unfortunately, in this country, race has been shown to associate with those types of factors that contribute to accelerated aging. And so this was a simple way of probing at why do we see poor generalizability across the data sets. And indeed, the ABC data set is more diverse than ADNI. ADNI is almost 95% white and very highly educated individuals. ABC is very similarly educated. However, it's almost 30 to 35% Black or African American identifying, which makes it considerably different from the ADNI data set. So this is just one factor to consider in trying to probe at the question of why we're not able to generalize the model. Beyond, of course, the known thing that it's a pretty small sample size that we're training on. And so ideally, pooling together much larger data sets would be a more promising avenue to continue this type of work. And going through, some of the bioinformatics analyses that I mentioned, the machine learning model, in this case, the ElasticNet, is able to identify various CPGs by, assigning weights to these CPGs, and we selected the ones that were given a non-zero weight. So we're able to pick out those CPGs from the model, and then use a platform or a library called KYCG, or Know Your CG, which is very similar to the KEGG database or KEGG pathway analysis that some of you might be familiar with. And so using this package, we're able to link the CPGs of interest to target genes that might be within, say, 10,000 base pairs of the location of that epigenomic marker. And using this, we identified a gene set of interest of genes that are located within this genomic proximity to the CPGs that we identified as interesting using our machine learning model. And finally, we used a package called Enricher to perform pathway level analyses. So not just genes, but what sort of biological processes, molecular functions, or cellular components are these genes involved in that could give us some insight into what are some of the biological underpinnings of the changes that we're observing here in the hippocampus and in aging. So basically what we did, which, you could argue, this is basically a sort of a proof of concept here. You could argue, well, you had very poor generalizability, very poor test correlation in your model. How do you justify doing an analysis like this? This is sort of basically, I think, part of doing all of these analyses at the CPG, gene, and pathway level. What we have noticed is that we see some consistent findings across all three levels of analysis, which gives us some indication for future exploration. It's basically generating a new set of hypotheses that we can test in future experiments. So again, this is all very exploratory work, but basically it's kind of proposing a new approach or way of thinking about these types of analyses, and proposing a framework of machine learning that we can apply to do these types of analyses, hopefully with larger data sets in the future and with greater rigor. So with that caveat, I'll continue and share some of these interesting results that we found. So of the CPG sets that we acquired from the ADNI trained and ABC trained models, we selected the top 25% based on the absolute value of the weights or the coefficients assigned to these features, and looked at the intersection. And of which we found this one CPG that came up in both of these lists among those top 25. And this is interesting because this CPG is located on chromosome 6 within the TREML2 gene region. And I'll talk a little bit more about TREML2 in a second. But what we know about this CPG or this epigenetic DNA methylation site is that methylation at this site is correlated with hippocampal volume significantly, modest correlation, but significant. And methylation at this site also correlates with DNA methylation levels in the medial temporal lobe. So we actually queried an external database that was looking at methylation in the brain itself, not in blood, and noticed that methylation correlates between blood-based measures and brain-based measures at this site. So that's significant as well. So I'm going to jump ahead a little bit, actually, talking a little bit more about TREM-L2. TREM-L2 is part of the TREM2 family, which unsure if you're familiar with it, but TREM2 has been identified as a gene that's, very involved in atypical Alzheimer's disease and neurodegeneration. So TREM-L2 is part of that family. It's not the same thing, but it is part of the family. And what we know is that it does have some relation with AD risk. It has some relation with white matter hyperintensity volume. But there's still a lot of questions about how these things relate to each other, and whether, TREM-L2, we think it's protective for AD risk, whereas TREM2 is actually a risk factor. So there's a lot to be learned about TREM-L2 and its role in Alzheimer's disease and neuroimmune response. So this is an interesting finding. Jumping back to the previous slide, one sec. The reason that we did a CPG level analysis and then a gene level analysis after linking the CPGs that were identified as significant from the machine learning model is there's a lot of collinearity among the CPGs in the epigenome, right? So we might have 900,000 CPGs, and, let's say maybe even many of those CPGs are highly correlated with each other. So the likelihood that even after feature selection, any given machine learning model will select the same CPG twice is very low, because it could just drop out one of the CPGs in favor of another that is very highly correlated with it or is located in a similar region on the genome. So instead of looking at that granular level, if we go one step up and look at the genes that are associated with those CPGs of interest, the likelihood that we find converging genes is a little bit improved, right? So this helps us perform a higher level analysis than the CPGs themselves, which are highly collinear and unlikely to pick the same one in any machine learning based analysis. So in this gene level converging analysis, we again identified TREM-L2, but also another one called BICL11B among these genes. And the reason why I picked BICL11B to talk about further is that it is known to be associated with differentiation in the hippocampus and the neocortex, which are two regions of interest for us, especially when considering atypical Alzheimer's disease. So while this gene has been, implicated in neurodevelopmental processes more than neurodegenerative processes, this is something of interest for us to explore as to how BICL11B might be contributing to neurodegeneration or aging, and not just neurodevelopment. It looks like there is a question in the chat, so let me try reading that. It's not popping up. One sec. All right. Thank you, Yashar, for your question. Have you considered exploring handcrafted features? For example, DNA experts suggesting features relevant to the task instead of using automatic feature selection, maybe a combination of both, shortlisting of features by domain experts, and then using automated features for further selection. This is a really great question. And I think this is exactly what we're looking at for the next step of this type of analysis, is to look at genes of interest. So, using literature and selecting CPGs that are perhaps already known to be associated with genes like ApoE4 or TREM2, the SOD genes, the APP genes, amyloid precursor protein. So we can look at epigenetic regions that are known to be regulatory of these gene networks, and use various other pre-existing libraries that can help us perform more biologically relevant feature selection. Absolutely. So this is something that we're hoping to continue to work on next. But absolutely, that is a great suggestion, that using some kind of biologically interpretable feature selection method rather than purely quantitative might be a good approach. So thank you for that question. Great. So just to recap, we looked at these two genes that were emerging as significant or interesting genes in our analysis. And this is very exciting for us to basically generate a new hypothesis that these two genes are potentially linked to neurodegenerative processes in Alzheimer's disease, especially in the hippocampus. And this can be further validated with more rigorous approaches. Finally, just to cap it off with that highest level of analysis, which is the pathways. So genes and regulation of genes is involved in various types of biological and molecular and cellular processes. And so we were able to use that enricher database to associate the genes that were converging that we identified and see what types of pathways they're associated with. And I want to point out these three that seem to be related to neurodegeneration, in particular apoptotic signaling, acute inflammatory response, and regulation of neurotransmitter secretion. So these are, three concepts that we are thinking are pretty important in neurodegeneration. And so what this shows us is, indeed, we see this converging evidence that this approach is helping us identify pathways that are implicated in the disease mechanism. So in the future, if we have larger data sets as well as machine learning models that perhaps capture more relationships among the data and not just in a linear fashion, so able to use deep learning, for example, we might be able to generate even stronger hypotheses and propose drug targets for validation. So in conclusion, in this work, we demonstrated that blood-based epigenetics reliably predicts age, as we know, and modestly predicts hippocampal volume. We noticed that the feature selection approach improves machine learning in the context of epigenetic clocks and epigenetic score development. And our converging epigenetic signatures from training models on two separate data sets implicate two genes, namely TREM-L2 and BCL11B, which may play a role in neurodegenerative pathways. And of course, as I mentioned, the caveat here is that we need validation in larger and more diverse samples to assess generalizability of these findings and of these models. And the work output here is a chapter in my dissertation, and I have code available on my GitHub as well. So to cap this off, we learned that blood-based epigenomic markers reliably predict age, and methylation at these two genetic sites might be key mechanisms to explore and validate in future studies. Just very briefly on extended applications and future work, we already had a couple of fantastic questions in the chat and live that probed at some of the things that we can work on next, which is really great. And just to add to that further, going back to AIM-1, we can extend the CoMeT framework, the Cortico-Medial Temporal framework, to not just look at structure, but also look at neuropathology as measured by something like tau PET or even amyloid PET. Tau is of greater interest since we know of its association with selective neurodegeneration. And so, we can extend the framework to use a tau PET in these cortical and medial temporal regions, as opposed to just looking at structure. And this would make this work more specific to Alzheimer's disease rather than somewhat general, where we might have confounding effects of TDP43 in diseases such as late or FTD. So this would help us focus the work and make it more specific to Alzheimer's disease. And similarly, so that's kind of that's kind of upstream of structure, and this half here is downstream of structure, right? So we could even extend CoMeT to look at a cognitive version of CoMeT and see how what is the axis of atypicality in terms of more cortical presentations that involve dysexecutive or lobopenic variants, where things like executive and behavioral function are impaired, or language, or even posterior cortical atrophy, where we have greater involvement of visuospatial loss of function relative to memory. So, both upstream and downstream of structure, which is what we looked at here in this work, we can generate a new indices that help capture that axis of atypicality on these levels. As I mentioned previously, we can also use more advanced and explainable deep learning networks. One suggestion in the chat was to perform feature selection using, experts and who have information about the relationships between these CPGs. And one thing that we can do is actually use a layer-based approach where we associate the gene CPG layer with various pathway layers and look at the association of the genes with various pathways that we're interested in, which would help capture more information about how these things are interacting with each other and not assume that they're operating independently. So this is one framework with which we could do that. We could also use graph-based reasoning, where nodes could be defined as CPGs or genes or even pathways, and edges could be drawn between and among these things to capture the likelihood that one CPG is linked to methylation at another CPG. Similarly, that one CPG is regulatory of a gene, or that a gene is involved in a pathway. And you can see that there's so many levels with which we could define such a graph. But using advanced methods such as this and focusing on keeping it explainable and keeping it biologically interpretable will be really helpful to generating clinical insights using these machine learning and deep learning methods that can be used by both clinicians and scientists who are working on therapeutics or on disease management for Alzheimer's disease and other neurodegenerative disorders. So as I mentioned again, dataset diversity is really important to consider. I would say that the genetic field is definitely much more advanced in terms of incorporating multi-ancestry into their analysis. So naturally, we would like larger data sets to improve our model performance and capture more complex associations. And if we do have increased sample diversity as well, we might have to incorporate methods that account for multiple ancestries as we develop our models. And this has been done in genetics and in statistical fine mapping methods that identify causal variants from a genome-wide association analysis. And very recently, there has been work started to develop multi-ancestry epigenetic clocks as well that account for genetic distance among individuals. So these are all sort of works in progress for the epigenetic field, but there's a lot to be learned from other omics fields that have already implemented these types of frameworks in their analyses. And finally, the ultimate goal of all of these things for me personally is to really unpack the associated biological mechanisms underlying aging and neurodegeneration. So we can use standard GWAS or EWAS, perform some kind of fine mapping and understand among the most significant CPGs or genes that are identified using these statistical approaches, which of those significant features are actually causal of the disease. And we can integrate functional annotations and epigenetic modifications to help us understand causality and not just association. And of course, colocalization analysis, which helps us relate different types of omics data to each other. So we can relate methylation with expression, gene expression, and better understand not just the DNA sequence itself, but how it's actually manifesting in the body. And with that, I'd like to thank the members of my lab and of the various centers that I was grateful to be a part of at the University of Pennsylvania. So the Bind Lab and Patch Lab, which were my two homes, broadly the Penn FTDC's the Frontotemporal Degeneration Center and the Alzheimer's Disease Research Center. I'd especially like to thank my two advisors, Corey McMillan and David Wolk, who were just fantastic advisors and always supported me and encouraged me to think outside the box. I also want to acknowledge the private foundation support that I got from the Penn Institute on Aging, as well as federal funding support from the National Institutes of Health. I was lucky to also be on a T32 program for translational neuroimaging and Alzheimer's disease, gracefully funded by the NIH. Very grateful for all of these people here on the slide, as well as Pulkit, who is leading this seminar and encouraged me to present my work here, who is also a colleague at Penn. So thank you so much to all of these people and to all of you for spending time with me on a Saturday and listening to my talk. I really appreciate you being here.
Well, that was a great talk, Lasya. A lot of new things for us to learn, and I think there were some really nice questions as well. Yeah, we are a bit over time, but if someone still wants to ask any question, we can always hang around a couple of more minutes. I don't see anyone asking any questions. But I think we had a nice discussion with all the questions we had, and
Yeah, for sure.
very thoughtful and nice presentation. So I think the recording will be available sometime in the next couple of days if someone wants to revisit the lecture slides or reach out to you. Yeah.
Awesome.
Thanks a lot for the presentation again. Yeah, great talk.
Thank you so much. And yeah, thanks everyone. Feel free to reach out to me by email as well if you have any questions. And feel free to rewatch the talk as well at your convenience. So thank you, Yashar, for your questions as well. Thank you.
Yeah, thanks a lot, Lasya. Thanks everyone for attending. Bye.